In this slide presentation, we're going to go over the integumentary system models and slides that are used in the A&P1 lab. Next slide. This is one of my favorite models because it shows you three different areas of the body and how the skin changes in those different areas. So like this first section is showing you the palm of the hand, okay? This middle section is showing you the axilla, and then this last section is showing you the scalp. Okay? Now, one of the first things you want to focus on is right up here, okay? And this is your epidermis, okay? And then all of this is the dermis, and then down here is the hypodermis. Okay, so I'm going to just kind of abbreviate it a little bit. So epidermis is up here, and then your dermis is this section, and then the hypodermis is that section. Okay? Now remember that your dermis can be divided into two sections. I'm going to draw an imaginary line right here, okay? And then this portion down here, let's actually make this a little bit smaller so I can write off to the sides. So this upper portion, right up in there, that is going to be the papillary layer. And what you want to remember is that that has areolar connective tissue in it. Okay? And then this section of the dermis is the reticular layer. And that is dense, irregular, so dense IR. Okay? The epidermis up here, you will remember, is supposed to be stratified squamous epithelium. All right, remember that the basal layer where you're going through mitosis, those are all going to be actually cuboidal in shape. And then the apical surface, which is the functional surface, that's where it's going to get truly squamous. All right, now with the integumentary system, with your epidermis, we actually discussed the epidermis in terms of 5 layers, 5 strata. Okay, S-T-R-A-T-A, which is the plural. The singular is stratum. Okay? Now, right here in this line, that is your stratum basale. Okay? Let's actually mark it kind of over here. So that is basale. All right, then this layer in here is going to be spinosum. Okay? And then this is in C. I'm just going to do the letter G because I'm running out of room to write, but that's granulosum. And then A, I'm going to do the letter C for corneum. Okay? Now, I want you to come over here, and in the palmar area, you'll see there's another whole layer, and that is lucidum. You only have stratum lucidum in thick skin, like on the soles of your palms and your hand. Okay, the palms of your hand, the soles of your feet. Now, if you look at the layer of A, you'll notice just how thick that stratum corneum is. It's extraordinarily thick in comparison to how much thinner it is right there. Okay, so thick skin has very thick layer of stratum corneum. All right, so let's start focusing in on the papillary layer. Let's actually clean this up so that we can see it a little bit differently. So hold on just a second. All right, so now that we're ready, let's focus in on the papillary region of the dermis. So this is the papillary region. And again, you're dealing with areolar connective tissue. And the areolar connective tissue is important because the epidermis is avascular, which means it doesn't have a blood supply, it doesn't have oxygen. So you need the blood supply in the papillary region, which is areolar connective tissue, in order to get oxygen to your epidermis. Notice right here the blood supply, and then you've got the capillary bed that comes up into this area. Now these little bumps, right here's one, there's one, those are the dermal papillae. The word papilla means nipple, so they look like little nipples, okay? And they go up, and the epidermis kind of locks around them, okay? And that areolar connective tissue gives it lots of space for blood vessels, and then also things like Merkel cells, okay? Excuse me, Meissner's corpuscles, not Merkel cells, Meissner's corpuscles. So that is a Meissner's corpuscle, okay, which is a light touch receptor. Another touch receptor you'll see are the Pacinian corpuscles, okay? Now, let's look at some of the gland tissue that we're dealing with. We have two different kinds of sweat glands that we're going to be working with. Eccrine and apocrine. Okay? Eccrine has its own duct. Apocrine shares with the hair follicle. Okay? And then eccrine is the one that you use for thermoregulation, or big T. Apocrine is our sex gland, basically, to keep it short. Now, the eccrine gland is right there, okay? Now what you will notice is you've got the base of the gland right here, which is going to be a coil of tubes, and then this is going to come up with its own duct that's going to keep on coming up, and then it's going to go around, and it's going to have a pore that comes out to the surface of the skin, okay? The apocrine gland is number 2, okay? And that's right here, okay? Again, it's got coils all in here. All of this is one single gland, and then its duct comes up here and it shares it with the hair follicle, okay? Now we have, actually let me label the other sweat gland, the eccrine. There's another eccrine gland right there, and you can see it's got its own duct coming out to the surface, okay? Now, this gland right here is completely different. This is a sebaceous gland. All right? It also empties into the hair follicle, just like the apocrine gland did, okay? But notice the structure. These to me look like saddlebags on either side, okay? Whereas the eccrine is a coiled snake, the apocrine is a coiled snake, just like the eccrine, all right? And then it empties into the thing up there, okay? Let's see. Another structure to look at is right here, and this is your erector pili muscle. This is not for touch. What it does is it moves the hair follicle, making the hair stand on end, so don't get that confused. We do have another touch area down here, right here, these yellow nerves that are coming around the bulb of the hair root. This is your hair root plexus. There's a couple of other names for it, but we're just going to stick with that one for right now. The hair root detects if the hair moves, and then it sends a signal to the brain. Okay? I think that's the basics. Let's see, what else can I point out? The palm of the hand, right? The palm, which is over here, you saw an eccrine gland. Here's another eccrine gland. All right? Eccrine glands are everywhere in the body. They're all over the place except for a few select spots. All right? Over here, which was the scalp, there's your eccrine gland right there. All right? And then a sebaceous or oil gland right there. Now this is the armpit. The armpit in the anogenital region is where you find the apocrine gland. So in the axilla, you will find apocrine glands, all right, coming up here. If we had a bigger model, you would also notice that there are eccrine glands and sebaceous glands present as well. All right, I think that about covers it for this model, so we're going to move on to the next slide. All right, so now we are looking at the skin. This happens to be a scalp slide. This is one of my favorite slides to look at. All right, scalp, it's actually a bald head. But for some reason, those are some of our better slides in the lab. And this one is at 100 total magnification, okay? And because it's at 100, we can see the epidermis here, okay? And then the dermis here. If I dropped this down to 40x, then we would actually be able to see some of the hypodermis as well, but we can't really see it at the moment, okay? So what I want you to focus on is the imaginary line right here between the papillary region and the reticular region. So this here, okay, that is the papillary region, and this is the reticular region, okay? And again, the papillary region is where you're going to find the areolar CT, whereas dense irregular is going to be found in the reticular layer, okay? When you're looking at the connective tissue here, what you want to focus on are the collagen fibers. So the collagen fibers are these pink lines here and here and here. And what you'll notice is up in the areolar region, these pink lines are very thin and they're more spaced out, and you can see more space, white space between them. And then down here, the collagen fibers are much thicker, all right? So the major difference between areolar and dense irregular is the thickness of the collagen fibers. They are laid out exactly the same way otherwise, okay? It's solely how dense are the collagen fibers. Now, in the areolar, you want the fibers to be thin so that you've got lots of ground substance so that you can have diffusion going up to the epidermis. Down here in the dermis, you don't have to do as much diffusion. What you really need to do is keep the skin from pulling apart. So you put in lots of collagen fibers going in multiple directions, dense irregular, which means it can resist pulling in that direction, and that direction, and that direction, and that direction, and that, and etc. So all different directions, okay? Now, within the dermis, you're going to find lots of other things embedded. So immediately what I can see is the stratified squamous of the epidermis. This is the hair root, and I can see the extension of stratified squamous coming down, lining the interior of my hair follicle, coming down here. Then I can see the saddlebags over here, which are my sebaceous glands. So I'm just going to put a little line over there, and I'm going to write sebaceous. Okay? Now down here, I can see a little bit of an eccrine or apocrine. No, it's going to be an eccrine gland, all right? An eccrine gland down there, lined with simple cuboidal epithelium, okay? Now if I look at this sebaceous gland, all right, what I notice are all these little circles, okay? And if you remember, sebaceous glands are going to be using holocrine secretion, which basically means they explode in order to release the oil, okay? And then the oil goes up the hair follicle and then spreads onto the surface of the skin. Now because it's an oil, right, you've got dead cell fabrics and oil mixed up in it. In order to do that, you can only rely on stratified cuboidal epithelium, because these cells down here need to be doing mitosis so that you keep making cells so that the ones at the edges can explode and die and secrete sebum, which is oil and cell particles, up to the surface. So that stratified cuboidal is important in order to accommodate the function of this tissue, because remember, form goes with function with tissue type, okay? The sweat gland down here is different. It's a simple cuboidal, and we'll eventually see a blow-up of that so that we can see it closer up. Let's actually move on and see if we can see a better picture of that. All right, good. So this is the same slide as we saw on the previous page. It's been moved, the focus has been moved down to the right where there is a beautiful eccrine gland. Okay. This is the same scalp, all right, and it's at a higher magnification now because I can tell because I can see the tissues better. So this is now at 400 total magnification, okay? Now all of this section right here is adipose. We're getting down to the area where you're closer to the hypodermis, so you start to see more adipose pushing up into the dermis. Eccrine glands will be found throughout the dermis. They sometimes are very close to the hypodermis, and sometimes they're up towards the more superficial edge of the dermis. It just kind of depends. Again, up here, you can see the lovely collagen fibers going in multiple directions. Okay. Now, I'm going to zoom in onto the eccrine gland. Now, remember that sweat glands are not cut in a single dimension. If this was a three-dimensional shape, it would look more like some kind of like long tubular snake thing and then go up like that, okay? Because you cut in half when you take a slide, what you do is you get to see all of these little pieces here. They're cut. Let me actually get a difference. So like right there, that's cut, and that's cut, and that's cut, and that, and that, and that, and that, and that. And that's what you see over here. So that each one of these little tubes is actually one circle of the cut single tube. So this is all one gland. All right? This is all a single gland. Okay? So now I want you to focus on one of these cuts. And you'll see the lumen of the tube right there. And what you'll notice is that it is lined with simple cuboidal epithelium. So let's zoom in even more. Now it's a little blurry, but I can draw on it, and you can see that they are lined with one row of cuboidal shaped cells. So this is simple cuboidal epithelium. All right, doesn't need stratified. It's not destroying itself. It's not producing insane quantities of sweat. It's just producing kind of a moderate quantity. So one row of cuboidal cells is sufficient for its function. All right, and while we're here, we can also take a closer peek again at adipose tissue. So you can see that it's got these chambers around in here. All right, each one of these little dots here is a nuclei. Okay, and it's, again, very common to find tissues mixed up together. Anytime you're dealing with an organ, the skin is an organ, so an organ is going to consist of multiple types of tissue. So that's to be expected, and you should try to find and label absolutely everything that you can possibly see. All right, let's go on to the next slide. All right, so these are, again, slides from the same scalp tissue cut. All right, but what we're doing is we're comparing and contrasting the sweat gland down here, okay, specifically the eccrine, and the sebaceous gland up here, okay? And you can also see very nicely down here the shape, directionality of the various collagen fibers, which is kind of nice. So again, I have a single sweat gland, okay? And up here, I have a single sebaceous gland, right? This sebaceous gland is stratified cuboidal, and this is primarily, the sweat gland is primarily simple cuboidal. Now, if I have apocrine glands, they will sometimes either be simple cuboidal or stratified cuboidal. And then sometimes my eccrine glands might look stratified if it's not been cut completely straight across, if it's been cut at a funky angle. Like this one to me looks very much simple cuboidal, but that one, you know, could be a little bit misleading. You might think it's stratified when in actuality it's simple. Okay? but up here with the sebaceous gland, it should be very obvious that it is stratified, cuboidal. Okay? Now on photomicrograph slides, I'm not going to ask you to distinguish between eccrine and apocrine. I want my students to be able to know that this is a sweat gland down here, and that's a sebaceous gland, but we really don't have the time to focus on distinguishing apocrine from eccrine on a slide. I do expect you to be able to tell the difference on a model, though. All right, next slide. All right, this slide is focusing primarily on the epidermis, and this is going to be a palm slide, and I know it's a palm because I can see a stratum lucidum and a very thick stratum corneum. Because I can see the layers so well, I also know that this is 400 total magnification. Okay? Now, before we get started on that, we want to label this. Okay? You want to label everything you can in a slide, whether that's what you're focusing on or not. So this is my papillary region of my dermis, and it is areolar connective tissue. Alright, I have all these purple cells, alright, and I know that those are cells. They're going to be a mix of fibroblasts and white blood cells. I'm not asking people to be able to distinguish those on a slide, but know that the purple dots are the cells. And then the pink lines, alright, in here, those are collagen fibers. Okay, and then the white space would be the ground substance. And then also elastic fibers, but they don't use the stain to make the elastic fibers show up dark on this type of a slide because it would wash out the other things. But you should just know that they're there. Okay, so right here, the very foremost layer, okay, that is stratum basale. And what I notice is that I can distinctly see the cells, their geometric shapes. The dark purple is the nuclei, alright, so that's a nucleus. And then I can see a little bit of like a white layering, a white layer between the cells, okay? And then this layer, that is stratum spinosum. Okay, and what I'm noticing is that my cells are starting to flatten out a little bit, but I can still kind of see them fairly well. Now I have this layer here, which is staining darker. In fact, it looks like there are little spotty grains in them, and that is my stratum granulosum. Alright, now remember granulosum has more keratin in it. So the name of each one of these cells in my epidermis, no matter where it is, so here, 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 those are all keratinocytes. Okay, so one of the features of these epidermal cells, the stratified squamous cells, is they have a lot of keratin in it. And you are increasing the keratin. So spinosum is increasing keratin. Granulosum is increasing a lot more keratin. Now, keratin absorbs stain more readily. Okay, so my granulosum layer stains darker because of the increased amount of keratin. Now I get to this lighter layer, which is called lucidum, alright, which means clear window. And it's a lighter color, and you're like, why is it lighter if there's more keratin? Well, you're putting more keratin in, but as these cells are getting ready to die, they're also becoming more lamellated. They're increasing their glycolipids. Their cell membrane is becoming very tough and rigid as they're ejecting the organelles, and it actually becomes harder to absorb stain in the lucidum, so it becomes a little bit lighter in color. And then, again, it starts to absorb stain more and the corneum. And you're like, well, why does that happen? Well, the corneum is dead. So when you've got a cell that's dead, its plasma membrane, cell membrane, begins to break down, which means it can absorb the stain again some more. And that is why the lucidum is lighter than both the granulosum and the corneum. So that concludes this presentation on the integumentary system's models and slides.